Good morning, everyone. This is John Hierdink. I'm the managing member of Tribe Public. Thank you for joining us today. We're excited to have one of our events here with an esteemed colleague named Jeffrey Meckler. He's CEO of Indaptus Therapeutics. If you don't know Indaptus Therapeutics, it's a very exciting company in the immunotherapy space, and thank you for your attention. They trade on the NASDAQ symbol INDP. And we're excited to have them today. And the title of the presentation is Immunotherapy Reimagined, if I didn't get too tongue-tied. But very excited. I've been spending a good deal of time with Jeffrey the last couple of days. As you know, many of us have seen me as well physically out at live events where Jeffrey has presented on the Tribe Public Network in tribe cities such as Newport Beach, San Francisco, in the Phoenix area, in Scottsdale, Peoria, and so on, and Salt Lake City the week before. And we're looking to getting them out across the country as we move forward. Please note, Tribe Public is a platform that's here to serve all. And in this case, we invite you to continue to stay aboard with us. And through our wish list process, send us the names of companies at our Tribe Public website, TRIBE Public, PUBLIC.com. It's 24/7, 365. And send us those names of leaders of companies that you care about, private, public. Most of the companies we've had on here to date have been NYSE and NASDAQ companies, but we're open. And, you know, as many of you know, we've actually had a Nobel Prize winner in medicine on here. So we're here to learn and learn with you and hear from you. And if you'd like to see Jeffrey in your city after this, let us know too, and we'll see if we can organize a nice luncheon event like we had yesterday and the day before. Thanks again for your attention. Also note that I am a registered investment advisor in California, run Vista Partners. That website is vistapglobal.com. Please look at our disclaimer language on both, as we are currently shareholders in Indaptus and serve as an advisor. So again, let me introduce Jeffrey Meckler. He's going to take us through a little bit about his background to start, and then go into his PowerPoint that's up visible now. And then we'll have a Q&A session after that. Remember, if you haven't sent in your questions yet and it comes up, send us through the Zoom chat feature during this. We'll do our best to get it addressed on this presentation. Should be done in less than 25-30 minutes at this point. And excited to have all of you on here. And note that we will be publishing the video of this event at our Tribe Public YouTube channel afterwards. So if there's anything that you think you may have missed, you'll be able to catch it there. And probably by the end of the day, I'll have it published there. So thanks again. Jeffrey, tell us a little bit about yourself.
Sure. And thank you, John. And I just want to thank everyone in the Tribe Network and everyone watching for giving us your time. My background, spent about 17 years with Pfizer after graduate school, and worked in a whole variety of things at Pfizer over my career in different divisions and different groups. Left and actually went into an investment fund. Did that for a couple years, worked for a variety of investors on small and large companies. And ended up getting into a few smaller companies. Have been CEO of, I think, 5 companies now. And what I'm very proud about in my background, and actually with the whole team here at Indaptus, is we get things done. This is a company that was formed just a little over 2 years ago. Dr. Newman, who's the inventor and founder of the Decoy platform that I'll talk about, and actually is really the one who is reimagining immunotherapy. He had a small company. It was just him. He had been working on this idea for a while, and it was ready to move forward, put together what's called an IND, an investigational new drug application, which is an application for the FDA to let you put this in humans. We formed the company, we raised $30 million, and we were off to the races. Last year was spent primarily doing things like manufacturing the material, filing the IND, going back and forth and trying to get the sites and get a clinical research organization all put together so that the trial officially initiated right before Christmas last year. And we started dosing our first patient in the first quarter. I think March is when we press released. And this is exciting for us because this is a whole new modality. And so as I get into the presentation, the slides are actually on our website. This was the same slide deck that we used at an investor conference a couple weeks ago. I'm not going to go slide by slide and go through everything. I'm just going to kind of tell you the concepts here and then show a little bit of data of why we believe this approach will work. One thing I do want to tell you is I will be making forward-looking statements. And as always, please see our filings at Edgar, companies, the SEC Edgar system, because that's where we'll keep the latest information. So just a really quick, broad overview about what, from an investor side, what Indaptus is. We're really developing a new approach to systemically administering anti-cancer, antiviral immunotherapy. And what's exciting is this is a whole new modality, and we've gotten into the clinic. We got through our first cohort. We've started our second cohort, and we now have getting to some data, and we're starting to see we're biologically active. And it's now really the development, the components of figuring out what's going to be the right dose going forward. How are we going to do the multi-dosing? And I'll go through our concept of why we think that's the direction to go. But this technology is really a platform that's allowing us to do a whole bunch of things, not just in cancer. But this talk will be really focused just on cancer. On the right side, John always is pushing me to make sure I talk about the stock. You know, we do have significant inside ownership. If you go on to Yahoo or the others, you see that number much higher. The 70% is just the management and directors. We have a couple other 5% or more investors. So there's not a lot of float out there, but it's an exciting stock, so it has some variability. But I'll let the stock people worry about the stock. I want to talk about the science, the technology, and why this is so cool. So this technology is based on a platform of engineered bacteria. Simply put, we engineer our bacteria, we then process it, and then use that as the platform to create a multi-package of immune agonists. So really simple is, our concept is a single pill, or I shouldn't say pill, a single approach is not the right approach in going after immune benefits. The immune system is complicated. There's the innate, there's the adaptive immune system. There's a whole variety of components and cellular pathways. And when cancer forms or develops, it's actually trying to hide from multiple things. And so you really need something that really engages the whole immune system. Now, what has happened, and this is where the reimagining immunology is coming from, is we have scientifically gone down an approach saying if you turn on the whole immune system, bad things will happen. And so we've really looked for the last couple decades in immunotherapy for that silver bullet. If we make this one little switch or this one little change, that will work. Well, what we've seen is the best immunotherapies only work in a fraction of advanced cancers. The reason is it's more complicated. Biology is more complicated. So what we develop is this package of immune activators that reset the immune system. And we do this through our engineered bacteria. So we have shown single agent activity. More importantly, we've shown tumor eradication in synergies with existing models. And we really have seen kind of a safe approach. We are able, I'm sure during the beginning of COVID, people heard of cytokine storm. And this is where really the immune system gets overexcited, overstimulated, and that creates a whole bunch of other problems. We didn't see that in our preclinical, and we haven't seen it so far in our first couple cohorts here. So we're excited that we think we have something that can be administered safely, systemically, to reset and really activate the immune system. So let me kind of get into this concept. So current cancer therapies, and I'm outlining some of them here in this slide, and I'm trying to show what they do. So when you look at CAR T or checkpoint therapy or targeted antibodies, what you see in this depicted diagram is what they're doing is they're actually just going after one set of the immune system, one part, and trying to turn on the actors in that immune system to do something, whether it's T cells or B cells or gamma delta cells or dendritic cells. And this is all important, but what they only work in those areas where that's the one thing that will turn the table on the cancer. The reality is the cancers, as they evolve, and particularly the advanced metastatic cancers, is that it's much more complicated than that. You can't just do one thing. And what you've seen over the past few years is combination therapy. Very often you see lots of trials with Keytruda, anti-PD-1. It's a checkpoint inhibitor. It is the #1 selling cancer drug. It sells over $20 billion a year. It really is the backbone of Merck's cancer business. And you see all these combinations. Well, if we add this, can we get the response rate from 18% up to 25% or what have you? So we are moving slowly in the direction of saying, well, let's put a couple things together. So Dr. Newman, who's the inventor of this technology, his thought was, well, wait a second. Instead of going piecemeal, isn't there a way that we could really engage the entire immune system in a way and do it safely? And Dr. Newman, his PhD from Harvard Medical School, was actually a professor, went to Roche Institute, has been in a number of companies. But where he was drawing upon was actually thinking about how mechanistically the immune system and the innate and the adaptive immune system work, and how you develop this in a way that really is meaningful. So one of the things that Dr. Newman kind of grasped onto was this concept, and it really was the backbone of the first immunotherapy, and it's called Coley's toxins. And it's kind of funny how it got the name. William Coley was a sarcoma surgeon in the late 1800s in New York City at what's now Memorial Sloan Kettering. And one of his fellow doctors who didn't believe in any of this, named Ewing, called it toxins because he said, oh, these are toxins, these are no good. What Coley had realized was an observation that had existed for hundreds of years, which is people with solid tumors had seen a regression in the presence of an infection. And we really, back then in the 1800s, didn't understand how the immune system worked. And there's lots of science that has gone on since then. But the concept of spontaneous regression in the presence of an infection was what caught Michael Newman's thinking about, wow, there are ways that the immune system gets turned on when the body is trying to fight an infection. And in going back and looking at all of this, he realized that it's a package of immune agonists. And what better way to develop the right package than to go to Mother Nature and look at how this works in the biology. And don't forget, believe it or not, you have just as many, if not more, bacteria cells in your body as you have human cells right now. Bacteria has existed longer than humans. We have evolved together. So what Coley was doing was actually taking a mixture of killed bacteria and trying to figure out how to give it so that he could elicit an immune response. And it worked. I've seen tables that show, durable responses, 5 years out from people who had used the Coley's toxins, up in close to 50%. And so we now, over the past decade, or two have learned a lot about the immune system. We know what toll-like receptors are. We know NOD, STING, all these other components and how they activate immune cells. So what Dr. Newman looked at it and said, look, let's try to isolate what we want to look at, what we want to derive. And so we know on the periphery of a gram-negative cell membrane of bacteria that most of it is what's called lipopolysaccharide, LPS, and it's an endotoxin. It's actually one of the most potent immune activators. And so if you have a bacteria infection, this is what will turn your immune system on. Well, the problem with LPS is in any large dose, it's so toxic and it's really overpowering the bulk of all the other immune activators. So Dr. Newman was looking at it, and there is research out there where people have looked at just straight LPS or taking all the LPS out. But Dr. Newman hypothesized that you need some of that for the activity and the engagement, but you also need all these other agonists, all these other toll-like receptors. So he developed this concept. And so what we do is we essentially take a gram-negative single pure strain non-pathogenic. And non-pathogenic means even if you got this as a live bacteria, it couldn't replicate in the human body. This engineered bacteria requires an enzyme that humans do not have. But we take that and then we reduce by about 90% the endotoxin. So we're stripping away the really toxic component of the outside of the membrane. Then we kill the bacteria, and then what we do is we do this cross-linking, we do this structural work. And what this does is, and the name Decoy, is it's setting up these cells to be administered in an infusion so that the body turns on and activates as if there were an infection, but there's no threat to the body. You know, we're not actually giving an infection. And the concept was, if we can do this in a way that's safe and systemic, we can activate the whole immune system. One of the challenges when you look at immunotherapy in cancer is a lot of the therapy is directed at the tumor site. You hear about the microtumor environment. Well, the problem is that the majority of your immune system activity is outside that tumor environment. So by giving something systemically, you have the opportunity of actually pulling this together and having the whole immune system activate and engage efficiently. So what we do is we take this bacteria, we process it, it's then put in a suspension. And so the way the therapy works is a patient would come in and they get an IV and they would sit there for a little while, just like they get other CAR T, other therapies, targeted antibodies, checkpoints. It's a very standard way, it's a very regular way that people do this. So one of the things, and I'll get a little bit into why the science works, but we're excited in that INDP 020, which is Decoy 20, we're in the clinic. Within 2 years, this company was able to take this idea and all the preclinical work that Michael had done and actually manufacture it, file the IND, get the clinical trial up and running, dose patients, and we're now in our phase 1. Additionally, we are now looking at how we can make other therapeutics on this platform. And essentially what we're doing, think of it in a recording studio. You're at a mixing box, you're turning down one channel and you're turning up another. That's what we do essentially, is we're engineering these next generation therapeutics. And so they're right now in the development, discovery, and optimization and characterization. But we hope to eventually have a handful of these different therapeutics that could have different applications, whether it's maybe some work better in some cancers than others, in antivirals, even in vaccines. So again, our timeline as a company, we're very proud that we're in the clinic. We're excited because literally we are getting data every week, new data from these first set of patients. We're monitoring them. But I do caution you in that do not look for this first set in the single ascending dose to really send a signal in the sense of efficacy. This is just a safety trial. We're looking to find the right safe dose that we can give, hopefully weekly, in either later this year or in the first half of next year to start our expansion multi-dosing. After we do that and we determine safety in our multi-dosing, and maybe we see a signal or two in efficacy, we'll then start combination therapy, where in combination with checkpoint inhibitors and NSAIDs and other things, we have seen 100% eradication of tumors in certain models. And I'll share some of that with you. Just kind of looking at the time, I'm going to kind of run through a few things really, really quick. This is a slide that just shows on the left-hand side a comparison of untreated bacteria to 2 Decoy therapeutics. And what you see is the activity in anti-tumor cytokines. So the immune activity that we want to see is still relatively meaningful. It's similar, even though we've stripped away the majority of the lipopolysaccharide, which is driving toxicity. And on the right-hand side is a comparison as a table looking at single agent approaches, their anti-tumor activity relative to a decoy therapeutic. And what you see with the decoy therapeutic is that what happens there is we have a lot more activity than a single agent. It makes sense why these single agents are disappointing in a broad part of the market. It's because they just don't generate that type of activity. We have started our characterization work of Decoy 20. We do have, this was a part of a poster. It's on our website that we presented at the AACR, it's the American Academy of Cancer Researchers. And it's just showing that we do have these various components. It's a package of immune stimulators that we develop naturally through these engineered bacteria. So I have a number of slides here, but I really want to focus. This is a slide that is really exciting from our standpoint. This is a slide that shows you what we give the cancer to a mouse. So this is a mouse model. This is HCC, hepatocellular carcinoma, so it's liver cancer. And what you see on that top graph, a kind of light blue, is what we call the control. And that is just saying we give the mice cancer and this is what happens. And these are error bars. So you see the average in there of the tumor volume, and then you see the range within there. If you give them an anti-PD-1, which is essentially what the #1 cancer agent, selling agent in the world is, you see a shift. It doesn't, on average, grow as fast. It reduces the tumors a little bit, but they eventually still grow. And you see 1 out of 6 CR, complete response. That means 1 out of the 6 mice, actually the tumors regressed entirely. And the rest of them, the remaining 5 out of 6, it delayed growth, but they still grew. If you look at our therapeutic, you see that we're essentially the red line, and for the n equals 6 here, for the small number of mice in this study, they're really about the same. So by ourselves in this setting, we're about the same as the best cancer agent in the market. But the exciting part, and that wouldn't get you excited, that's already out, it's approved. But when you combine them, you get to see the 6 out of 6 complete response. And it's very meaningful in that all the mice, the tumors all regressed and were eradicated. Now, at day 91, 5 out of 6 mice were still durable. That means 1 of the 6 had cancer reemerge. Now, to understand, you go, I got asked the question once, well, 90 days isn't that long. Don't forget that an average field mouse lives only a year. So the lifespan of these mice compared to lifespan of humans and how the cancer works, they're very different. If you look at other companies and how they publish their immunotherapy data, you would see a slide more like this. Most companies only go out maybe 28 days. And they show that separation and they get all excited about, look at how much improvement we have. We go out much farther because we're really trying to understand, is the immune system doing what we want it to do and what we think it should do? Now, the other slide I want to spend a couple minutes on is this slide, because this is the holy grail of immunotherapy, and that's immunological memory. And think about it this way. You get a virus or you get a disease and you're sick for a little while, you get better, and now you have some immunity to it. It's the whole concept of vaccines and the idea that your body learns from the exposures it has. So one of the things we wanted to see was, with these mice with hepatocellular tumors, let's give them another hepatocellular tumor. They've cured the prior total eradication. I know some people hate the term cure in cancer, but they're total eradication of tumors. And we give these mice a hepatocellular carcinoma again, and sew it in. And we give them to them, on the left-hand side are the mice that totally eradicated.
Excuse me, it looks like we have lost Jeffrey here. He should be able to get right back in. I apologize for this. He might have some sort of internet issues there. The one thing I'd while we're waiting for him to get back in, is I want to point out is that the company has a relatively low float, about 8.4 million shares outstanding. And if you pull up on any of the services, it shows about 40% is held between insiders and large investors. So it's a relatively tight float overall by most people's look at the situation. And it's been volatile up until recently. Last week, if you look at the chart, it was establishing 52-week highs on a daily basis until our good friend Uncle Jerry from the Fed started speaking, and it seemed it chunked its way back down. Having said that, we're still up around these levels, about 83% year to date, which is not your average return on small microcap biotechs this year for sure, especially after the last 2 years of the sell-off, as many of you've been viewing the space as well. Jeffrey just texted me and said he's trying to get back in. And I'll see, hopefully we'll have him any second here. Remember, you can send your Q&A to me through the Zoom feature if there's anything on your mind currently, and or when Jeffrey gets back on, he can address. But it looks like he had some sort of technical issue there. I will say this: over the last couple of weeks, when we've had him out in these different cities and meeting, it's been overwhelming excitement about what he's potentially, hopefully, going to bring to humanity here. Hi, Jeffrey. I see you back on. Can you hear us?
I'm coming back. I apologize. I don't know what happened. The whole system just shut down here. So I'm on my phone now.
So just so you know, when you're off, I started talking about your float and how tight the insiders is, and also encouraging folks to send us any additional questions. So anyway, if you could pick up where you were and maybe finish that thought, and then we go quickly into Q&A. So.
Great. So what I was talking about was immunological memory. And so the concept here is that what we are able to show, and what you saw in that slide, and I'm sorry, for the technical difficulties here, but what we were able to show is that by giving the cancer, re-challenging to those mice, they were all within about 2 weeks, their immune system identified the cancer and were able to reduce and actually eliminate all of those tumors. So we saw immunological memory. And on the right side of that graph, I don't have it in front here, but you can go on our website to get it. You see when we gave that to naive mice, and what happened there was those mice, all the cancers grew. So it demonstrated that once we're able to find a way to eradicate the tumors with a decoy therapeutic, the immune system has learned how to respond and in the future clear that cancer. So it's a huge, so far, even the best therapies today have recurrence. If you have Keytruda and it actually reduces your tumors and works, the thing is you still have to, it's coming back. You don't know when, but it's coming back.
Yeah, and Jeffrey, you mentioned, I believe Keytruda is only effective in, what was it, 1 out of 5? Is that what it?
Roughly 1 out of 5.
Yeah.
So
it's still a $21 billion drug in 2022. Amazing, and it's helped so many. But even that, there's still a big opportunity to assist. And you said there's a failure rate after that. The second time, there's some effect.
We do know that these immunotherapies that we're doing now with checkpoint therapies and others, even the targeted antibodies, they work for a while, but they all have a developed resistance, and over time they don't work after that.
Gotcha. Thank you. Did you want to go on to sort of maybe your milestones and what you're looking for this year and cash position to get to those?
Sure. So the things that are going to be happening and what we've been doing with the tribe events is, I call it my If a Tree Falls in a Forest tour, is just trying to get out there so that people realize, because we expect to have data here in the fall. And this is going to be data from our first cohort, and we will get it out there at some point, whether it's at a conference or we'll do an R&D event or something. But what we're going to find out from this data is, how biologically active, how safe, and we're going to have a lot of data around there. And then we're going to be able to use that to determine when we go into our multi-dosing. And that's big for us, because going into the multi-dosing is where we think we will see some efficacy signals. And that will, hopefully start either late this year or into the first half of next year. So over the course of the next 12 months, we have a lot of potential data points coming through. On the cash burn, we burn about $1 million to $1.2 million a month. Mm-hmm. We had 19 million plus at the end of the second quarter. So we have enough money taking us out into what we believe the middle of next year. And so we'll have a lot of data coming between now and then.
Fantastic. One of the questions that's come up, it says, why do you think Decoy 20 shows single agent activity in some mouse studies and not others?
Good question. Someone who's looked at our website. Yeah, because in our non-Hodgkin's lymphoma models and whatever, we do nothing. It really has to do with every cancer is a little different. And we believe that in some settings, the package of immune agonists have to be different. And right now we have Decoy 20, which is basically taking the package of immune agonists on this gram-negative engineered bacteria and just turning down the LPS. We think by turning up other and turning down others and adjusting, as I used that analogy being in the mixing studio before, by doing that, will allow us to work in different settings. So right now, Decoy 20 works really well in some settings. It's as a single agent. If you look on our website, whoever, for that question, yeah, and you look at the antigen expressing breast cancer HER2 mouse model, you'll see 2 out of 6 mice had complete responses, single agent decoy. So it works better in some than others. That'll be part of what we find out when we do more human studies. And then as we learn more, we'll have other decoys that will adjust that package of immune agonists to work better in different cancers.
Okay, thank you. The next one is, if you just gave LPS as the most potent immune stimulator by itself, why wouldn't that work?
It's a great question. There are a lot of studies actually out there where people used LPS, and you can give LPS. It's actually allowed to be included into certain adjuvants and therapies. It didn't work. I mean, it's been tried. Synthetic LPS has been examined in a lot of studies. We believe it doesn't work because you need the package of immune. Again, it's the same issue as why a lot of these other therapies, it's only one agonist. It's only one pathway being engaged when you just use LPS.
Yeah. Switching to the stock side of things, one of your analysts, it's understood, and if you confirm this, updated recently, I believe, out of Maxim, and he has, I believe, a buy on you. And can you confirm the price target and how he came to that?
Yeah. Okay. So Jason McCarthy at Maxim covers us. I think we have a price target of $4. That price target was set when we were $1 something a share. It'll probably, we hope, gets revised here as we've come into it, and it was actually put out there before we had human data. So we hope that as we get data out here in the fourth quarter, that actually gets revised. If you look at, we also have H.C. Wainwright, and they have us at, I think, a $12 or $14 target price. You know, with little companies like this, it's really tough to say what's the value. You can look at comparison to others. You can't really make a P&L out 5 years and put a discount rate to it and say that's the value of the asset.
Okay, thank you. Well, it's good that you got some coverage, and that people are starting to pay attention to what you're doing. I think these events this fall could really bring further understanding, obviously, to you and to all, really, and investors. It's going to be could be a really exciting fourth quarter for you. Just one last question here that I'm seeing from the group says, why would you lower the dose in the second group?
Yeah, the reason is that we saw biological activity in the first, and there's a slide that shows we have a very broad therapeutic index in our preclinical models. When we go forward in the multi-dosing, the goal is really to give the lowest dose that's biologically active, giving the immune response, because we'll be giving it repeatedly. We believe this pulse prime, this giving it clears the system in a couple of days, give it the next week. That's going to be the best way to really reset the immune system. And just for the potential of other things that could happen, we want to go to the lowest dose to manage that. And so what it tells you is our first dose actually hit our activity that we were looking for from an immune response. So now we want to see if we lower the dose, will we get a similar response as we try to target it in? It shows us that the first dose was really doing what we wanted and was a lot closer to what we want to give in multi-dosing going forward.
Got it. Thank you. Actually, there is one more, if you don't mind. It says, are you worried that another company would just use a different bacteria to do the same? I guess worried about competitive advantage here.
Great question. No one else, our patents are for using gram-negative bacteria. We've never told anyone exactly which strain of bacteria we're using, but our patents are actually broad to any gram-negative bacteria. Now, that being said, other people have used oncolytic viruses in a similar approach. We don't believe those will be so successful because they don't really evoke the same kind of immune response. They're really more targeted at the tumor, the virus attacking it, and the immune response systemically is much more muted. We're the only people who are doing this type of approach, and our patents cover a much broader than just any given bacteria.
Got it. Thank you. Well, I think we'll conclude here, and I want to say thank you to all the tribe members that came in from around the world. I see a couple people that are up, I guess, a little bit later in UK and have joined us. Thank you so much. It's always great to see people from all over the world that come into our events. Note again that we'll get the video up of this event by sometime today when we get it up there at the Tribe Public YouTube channel, and then we'll be connecting everybody with our Tribe this week, which is our e-newsletter that goes out about 1:15 every Friday, and that will encourage you to see the video there and to get the latest updates on Indaptus going forward. And we appreciate you, Jeffrey, for joining us, sharing your insights, and invite you back, hopefully, as you're putting forth positive information about your trials, and we're excited to do that. And if you guys do want to see Jeffrey, remind you to use the wish list process. Ping me an email, whatever you want to do, just let me know. And if we get enough interest in your area, we'll approach Jeffrey and see if we can get him to travel out to see you. He's a little bit of a world warrior these days, and but we're happy to see if he'll fulfill those wishes. Thanks again. Remember, the symbol for Indaptus is INDP. And if you got any questions at any time, please follow up with me. And I'm happy to try to track down some information and or connect you to Jeffrey directly if we can find time in his busy schedule. So again, Jeffrey, thank you so much. Thank you, Tribe. All the best to you, and have a great rest of the weekend here that's starting pretty soon. All the best.
Thank you.